Hola, clase. I'm going to give this entire lecture in Spanish, so just hang on, you'll be able to get it. So in order to do that, I'm going to use the abbreviated form, and I'm just going to put one L at the beginning of the lecture and then one O on the end. So when we're ready to go, you'll see it. So L, DNA, is what we're going to be talking about here, and we're going to be talking about specifically what DNA polymerases are going to be doing. So this is DNA polymerases of any variety. So whether we're talking in vitro or in vivo, almost all these polymerases are going to work in basically the same way. So a DNA polymerase is going to come to a DNA strand, and it is going to be adding on to the 3' end of a carbon, of this carbon sugar. So what we got here, you can see, is a phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar backbone. We've got our nucleotide or base, the nitrogenous bases off here on the side, G, T, A, C. So an enzyme is going to come in here, and basically what should happen is a nucleotide triphosphate is going to come in and it is going to be added to this chain by the enzyme. So one of the questions you might want to ask is like, well, why would this nucleotide come in at all? That's a very good question. L-nucleotides will come in for a very particular reason, but I'm going to get to that a little bit later. So what we need to make sure that we have here is a nucleotide with a 3' hydroxyl group, and we're going to make sure that we have a new nucleotide with 3 phosphate groups. That's going to be really, really important. So this is going to be the site at which we are going to add, and these 3 phosphate groups are going to provide the energy. Remember that if phosphate groups are really, really negatively charged, so if I want to push them together, it's going to take a lot of energy, right? So when I have 3 negatively charged phosphates this close together, they're going to blow up basically when I snip one of these bonds, and that's exactly what's going to happen. That is going to cause a net release of energy, and it's going to allow the enzyme to push this phosphate group up into this hydroxyl group. And we are going to run a basic dehydration synthesis. Again, we're going to make sure that we're going to need a 3' end to that nucleotide O. My throat's getting sore. But again, why the heck did this T come in? Like, did DNA polymerase just think like, hey, I want a T, and then it came in? No, that's not going to happen. So let's not forget what's going on here. DNA is rarely single-stranded. So there is probably another strand of DNA, and I'm not going to draw the whole thing, but that is parallel to this, or if you're kind of a stickler for terms, it's going to be anti-parallel. There is going to be an A here, a T here, a G here, and an A here. All right, so it's really this attraction to this that is going to draw in the T. It's the enzyme that's going to attach the backbone that's going to covalently bond these two things together. One more important thing about a lot of DNA polymerases, and something that kids screw up oftentimes when they're doing reactions, is that the enzyme that puts these things together often has a magnesium ion embedded inside of it. Now, if you'll remember, the backbone of DNA is negative. Every single phosphate group is negative. So that positively charged magnesium that lives inside this enzyme is, did I get it? Is that my pointing to the enzyme? Yay! Yay! I forgot where I was. Right, so that magnesium inside that enzyme is going to allow this enzyme to interact with the DNA. So if I lack this magnesium, then the DNA polymerase a lot of times will not work because it won't be attracted to this growing chain, and so won't do what it's supposed to do. So in a lot of the labs that we're doing and a lot of things we're going to be talking about, whether it's in vitro or in vivo, a part of this chain is already going to have to be there. DNA polymerases can basically not start nucleotide synthesis de novo, which means, I don't know, newly, right? So they're going to require the presence of this hydroxyl group that was here. So that's why in the case of PCR, we have to buy DNA primers from the good people at IDT DNA. Or in the case in vivo, we're going to actually have an RNA, piece of RNA built here with a hydroxyl group there that's going to allow us to build off. So we're going to need kind of this 3' end. We're going to need a template strand so that we know what letter, so that the letter can line up and we can joinify, covalently joinify those things. We're going to need some magnesium for our enzyme. Our energy here is going to be in the triphosphate that we're adding. So the triphosphate that we're adding is going to be a D-ATP, a D-TTP, a D-GTP, and a D-CTP. Depending on what letter I'm adding, that triphosphate, 2 of those phosphates are going to break off, providing the energy for the reaction, and then it's going to turn out that the remaining thing is going to be the nucleotide, which will act as our raw material for the reaction. So what do we need to do this? When we're running PCR reactions, when we're running replication, we're going to need a 3' end, we're going to need a template, we're going to need energy, and we're going to need raw materials. So that's going to be in the form of our DATP, TTP, GTP, CTP, TTPs, which we can just simplify as DNTPs. And that's all, right? Oh, magnesium and an enzyme. Yes. Okay. Have a great day.